I think maybe it's a time to start the seminar. So thank you so much everyone for joining the webinar today, which is part of the single cell and spatial exploration group. I'm Aaron I'm from the BioTuring team, and today I would like to share with you about the ability to build the single cell atlas across different diseases and also including the healthy control as well. To provide one example today, I'm gonna use one example of the target called Nectin-4, which is recently an interesting target for the drug antibody-drug conjugate to demonstrate some of the functionality of the atlas that we can use. And from that, I can also give some basic example of how we can use the public single cell data, which is the curated data, with standardized QC and metadata to achieve this goal. Okay. So I think this is the quick agenda for today. So first, I would like to go overview of the single cell database that talk data have. So because the talk data will have more and more single cell that can cover a wide range of human diseases. So I would like to go overview of this database first, and then I will show how to create the custom atlas for a particular disease of interest using the talk data. And after create a custom atlas on a database, we have the option to export the custom atlas to a single cell analytic platform, which is we have BBrowserX Pro today, and to perform some ADC target validation. And for example, I mentioned Nectin-4 and one example that I use today. And after creating the atlas for the disease tissue or disease and the condition, and it's also important for the drug development that we can also have the way to look at like healthy normal control. So in this, we can use the same methods to create atlas for the healthy control tissue and profiling the Nectin-4 expression for off-target prediction. So for today's demonstration, we will focusing more on the usability of the platform, on finding the target and validating target, rather than the picture demonstration. So this is the overview of the single cell database talk to data with data curation here. So for the talk to data, recently we have the single cell data from different species. So we can see majority of the data come from human, and we also have the data come from animal model like mouse or non-human primate. And this is the number of cell and number of study. We can see there are 3.4 thousand study curated by our team. And also we will have the new version coming soon, which is will include more and more single cell data for you to explore different diseases and also increase the number of cell for particular. Sometimes like people would like to study some cell type that's very rarely is detected. So with a big database, you can able to make the atlas comprise of all of these cells into specific disease atlas or cell atlas to use for your particular purpose. And the talk data cover variety of human disease from cancer, autoimmune disease, neurodegeneration, heart disease, lung disease, and also infectious disease. And importantly, they also have the control, the normal control with that. Yeah. And this quickly show you how the data are curated, because one of the important thing is doctor data is the database that we curated the published data from publication, and it's very important to process the data in the same way to be able to compare between different data sets. And also I also mention later that in the new atlas we also do like best effect removal before we do any analysis. So this is after we get the data from the published database, we will, if they have the raw data, so we'd like to start from the raw data, and we run the same pipeline from processing, metadata curation, cell type prediction, and also for the QC to make sure that all this step is unified across different data set to be able to make the better comparison. Okay, and before we go into the ability of creating custom atlas, so I would like to go a little bit on the target that I use and the one example for today, which is Nectin-4. So Nectin-4 is the important target for the antibody drug conjugate or ADC development because this target is very selective for tumor cell. So Nectin-4 is highly overexpressed in solid tumor and a very limited expression on normal tissue, which is very important for the selectivity of the ADC target. And especially Nectin-4 is have a very rapid uptake and liposomal processing, which is very important for the efficient of internalization. It's very important for the ADC and also is clinical validated. So there one drug get approved, which is the enfortumab with vedotin, which is I think reimbursed to treat bladder cancer that confirm Nectin-4 as a druggable target for ADC. And recently, the expanding pipeline of development of the next generation of ADC with improving the linker payload and also configuration strategy to make the better ADC with Nectin-4. Okay, so that's the overview of target we have today. And one of these questions that usually I would like to ask before I get into any target is because they have different cancer type, and I would like to ask like which cancer type because Nectin-4 we use in the bladder cancer already. So besides bladder cancer, any other cancer type may benefit from Nectin-4 ADC development. So for that, we can go to the single cell data comprised of different cancer type, and then we can list on the cancer type on the database shown here, and then we just simply first overview by simply query Nectin-4 and see which cancer type that highly express Nectin-4, which is maybe a good target for the Nectin-4. So you can see in this case, you can see there cervical squamous cell carcinoma on the top of the list, and before that, you also have some basal like breast cancer carcinoma and also some other oral cavity cancer as well. So, but basically there are some few of the cervical cancer shown in the list here. And maybe I can quickly show you how to achieve that on the platform. So first, so this is the single cell database that you can see. We have different databases, but today we will focus on the single cell. And from that, you can filter for the condition. So I select all of these cancer that we able to get from the database. And from that, you also select the suspicion type of the cell. And now we can go to this custom T view, and then we can select the condition, which is we want to list all of these cancer type on this view. So then we can just search for Nectin-4. And then with the searching, you will able to sort that. So the high expression on the top and low expression on the bottom here. And you can see very clearly that cervical squamous carcinoma is the cancer that have the Nectin-4 highly expressed. And one of these was very quick way to screen for the Nectin-4. And for example, if I'm interested in targeting on the cervical squamous carcinoma and would like to create a single cell atlas for the cervical cancer, because I would like to cover all different types of cervical cancer. So instead of just using cervical squamous carcinoma, I can cover other cancer types as well. So for that, I can just simply go to here. We have the atlas function here. But before we create atlas, we need to kind of filter for the data to get the data that we want because then 3.4 thousand study here. So that I would like to first for sure, I would like to have a normal control. And then I need to have the cervical cancer. So I select on the cervical cancer and also subtype, and I would like to include the cervical neoplasia as well. to put into one single atlas, because later on I can show you after we create atlas, they have the metadata condition. From that, you can split the data into different subtype or different subset of cell, which is okay. And for anatomy, I would like to go to uterus, because sometimes people study cancer, but they're using blood sample. So I don't want to include as blood sample. So from here, you can see the study. There are 27 study that satisfy all of these criteria that we filter here. And now we can go to the atlas on the top data and then create new atlas. And they already have 1.1 million cells. That's the one that we filter, and 27 study. And then we can continue. And here I just give it a name. And then you can see here that embedding methods. So because we combining different data sets come from different study, so it's better to do harmonization to be able to kind of remove the batch effect come from different study. So they have the option to do BCA with harmony here, and then you just create, right? So the platform will process the data, then combine data, run a batch of fact removal, and then when it's done, so I have one example, exact example that I prepare to save some time. So you can see here, this is the atlas that created from the cervical cancer and also the normal control. And here you can see there are a list of 27 study that we combined before, and on the metadata also we combine like cell type, anatomy, and condition here as well. And from this, you'll be able to export into the BBrowserX Pro platform, which is more analytic platform for single cell data. So talk data will be more about searching and creating atlas, but for analytic analysis, so I recommend to go to BBrowserX Pro. And then so you can see we can generate the atlas will be transport into the BBrowserX Pro, and we save the folder under name talk to data atlas. And I create different atlases here with different number of cell. And for example, the one that I created is the cervical cancer versus the normal atlas. And also create some atlas beforehand, for example, the normal lung atlas, normal kidney atlas, just for the demonstration today. So I just go to the cervical cancer, and you can go to the condition here to take a look. So in this cervical cancer, and you can see there's some normal sample, there's some cervical adenocarcinoma, there's some cervical squamous cell carcinoma as well. So different condition, because I combine these conditions together into that lab. And then what I would like first, I would like to do is I would like for sure, because we would like interesting in Nectin-4. So I would like to query the expression of Nectin-4 for some across different subtype of cervical cancer and also normal. So I just simply go to the visualization and basic chart here, and align the bubble plot heat map here, and let's do Nectin-4, right? And you can see here. So the expression of Nectin-4 is different with different cervical cancer subtype and also with normal. So you can see normal is have the very low expression compared to, for example, cervical squamous cell carcinoma, which is validating the overview that we have just done on the Talk to Data platform. And also you can see beside that other like the cervical squamous neoplasia also highly expressed Nectin-4, but not the cervical adenocarcinoma. So I think that is interesting, because it's also nice because with the Nectin-4 is the target for more cancer, but not much expression in the normal control, which is important. And the next question that I would like to ask is, what is because this is over the condition, so on of the cell type. So what I interested is I would like to know what is the percentage of the cancer cell that express Nectin-4 in cervical squamous cell carcinoma. So I only focus on this subtype now. So for that, what I would like to do is I just subset this. So I just, like I mentioned before, you can build the atlas within different condition or different control. And then if you would like to study a particular subtype, you just simply subset that into a small subset and you can save that. So we could, so we might do, because I already subset one more time. Okay, and now you are working on the cervical squamous carcinoma only. And before that, I also do one more thing. I would like to select the gene that is with Nectin, the cell that is with Nectin-4. Okay, so you can query for the cell that is with Nectin-4, and depend on, you can do select the cutoff that you think that will be positive for Nectin-4. But for here, I select on the cell that is with Nectin-4, and I annotate that into a cell subset, sub3, for example. Nectin-4 positive cell. The purpose is this: we would like to understand the proportion of the cell that express Nectin-4 and the proportion of the cell that in the total number of the cancer cell. Or I label this negative. So now we annotate the two populations into the metadata on cell sub3. And now we can go to the cell proportion to visualize that. So because I'm interested in the uh, cell type, so you can see in this atlas, we collect 99 cell type here. And then you can do the proportion of cell sub3, for example. I think it's the same for one because I did it before. Okay. And now you can see, scrolling down, most of the cells that have a very few percentage of Nectin-4 positive cell. For example, like mast cell, we have around only less than 1%, and other cell like plasma cell is 1.5%. What you can see in neoplastic cell, which is cancer cell, let's show like 61% of the cell that express Nectin-4, and other 38%, the rest is not express of Nectin-4. And you can also consider there may be some dropout during the single cell analysis as well. And you can see beside that, there are some epithelial cell. So epithelial cell, there are two things. So Nectin-4 is attention molecule. So they also expressed in the epithelial cell, and also in the published data set. Sometimes like the author annotate cancer cell at the epithelial cell. So that's why you can see there increase in the percentage of the Nectin-4 positive cell in the epithelial cell. So beside that, other cells have a very low percentage of cell that have the Nectin-4 positive. So it is a very nice thing that if you use the Nectin-4 to target uh, cervical squamous carcinoma, it's more likely hit into the cancer cell population here. Okay. So for that, would like to go back to the slide here. So for that, we already create the atlas and do some basic cell proportion, but this only helped us to answer like which cancer subtype that highly express Nectin-4 and how many percent of the population of the cell that have the Nectin-4 expression. Okay. So I use the same analysis, and we can understand more about the normal tissue. So with normal tissue, so we can survey across the normal uterus tissue, and only you can see there's a very small percentage of epithelial cell that had expressed Nectin-4, and it is most of the other cells that don't show any Nectin-4 overexpression, so which is a very nice for the selectivity. And beside that, also we can also use the single cell platform to run some pathway analysis that can help us to understand more about the cell subpopulation that we're working on. For example, is here I compare the gene expression of the Nectin-4 negative population compared with Nectin-4 positive cancer cell. So we can see there the decrease in cell-cell adhesion, because it makes sense because Nectin-4 is cell adhesion molecule. But beside that, you can see also the decrease in the endocytosis and also vehicle-mediated transportation, which is shown that in the group of the Nectin-4 negative, they are less active of this endocytosis. Because and also because with this the kind of relative comparison, which is mean the Nectin-4 positive cancer cell have the higher endocytosis. So what does mean? Endocytosis is very important for the ADC. So if we target Nectin-4, it's more important for the positive cancer cell to attack the antibody. And beside that, for sure, we can do also differential gene expression to understand more about the two cell population. So if I compare the Nectin-4 negative compared to positive cell population to see any target that I can target during uh, the negative population, because with negative population we cannot use Nectin-4 to target for destroying the cell. So to compare that, so we can see there are some interesting target like CXCL4. So CXCL4 is the signaling that activate the cell proliferation, survival, and antibody anti-apoptotic signaling. So, and also there are some experimental drug like the ADM3 100, that is the small molecule that can target the CXCL4. So that is the maybe that is one of the suggestion that CXCL4 can be the target for the cell which is have Nectin-4 negative. So we can cover both Nectin-4 positive cancer cell with ADC and Nectin-4 negative cancer cell with the small molecule drug. Okay, so we use the same strategy to create the off-target analysis. So there are two ways to test the off-target analysis here. So I can go back to the talk to data platform here. One thing of the Nectin-4, people are aware that is Nectin-4 is highly expressed in the skin. So whenever we have some drug with Nectin-4, we should look to the skin first. So we also want to verify this here. So I have the selected here. So I can filter for all of the skin in the anatomy shown here. And then I can select the list here as the anatomy. And then I can query the Nectin-4 again. And you can sort. And you can see very clearly that the skin of the manus, which is the skin of the hand, that is more highly expressed Nectin-4. And for example, I would like to see what cell in the skin that highly expressed Nectin-4 is most likely to be affected by the drug targeting. So you can see keratinocyte is the one that highly express Nectin-4. So if we use Nectin-4 as a drug for ADC target, so most likely there we have some maybe um, side effect on the skin, especially on the keratinocyte here. And that is the one quick way that you check the up target. But there also the way that the more precise way to check the up target is you can go you can build atlas to for some particular tissue that you're interested to look into more detail. For example, here I have the atlas for the lung, so normal lung atlas open here. And this normal lung atlas, we can also able to annotate the cell into the Nectin-4 positive cell and Nectin-4 negative cell. And for sure, we would like to survey on the whole cell type here. So which is 222 cell type that I combined from the publication. And then we can go to the cell subpopulation here. And then you can query. So see multiple cell, they don't have any Nectin-4 expression, which is good. We will not do any off target on these cell. But when you scroll down, you can see starting they have some basal cell, which is 16%. They have the Nectin-4 expression, and also more on the epidermal cell and goblet cell, for example. So that will with the ADC for Nectin-4, so there will be some effect on the lung, and the main effect will be on the basal cell and also epithelial cell and also the goblet cell. So that is the one that we can check for the off target. And from this, you can do further detailed analysis on which cell type in which condition you can do more about like the progression of the disease with the disease atlas as well. Okay, so I think that is the pretty much for most of the thing that I would like to introduce about the atlas. So this is the kind of summary for the today club. So with the custom atlas we built on the single cell database, we are able to see Nectin-4 as potential target for ADC development, especially for the cervical squamous cell carcinoma. And the high proportion of the cancer cell in the cervical squamous carcinoma that express Nectin-4, which is very important for the ADC targeting, and have a very low or no expression on the normal tissue. And in the Nectin-4, in the group of the Nectin-4 negative cancer cell, you can see there are other target emerging when we do the differential gene expression, for example, CXCL4, which is maybe important for targeting using the small molecule drug. And profiling the Nectin-4 expression on different normal tissue and across the large number of cell type for better prediction of the potential off target, which is I quickly show you two examples using the single cell database, or you can build a custom atlas to look at the target expression across 222 cell type. Okay. And from this, I would like to introduce some resources that we have here. And I have also some survey that I can put here in case you can here. Okay. Yeah, I have a quick survey. It would be very nice if you can, if you have any disease in your interest and any particular difficulty or any concern during the data combination for building the disease atlas. So please feel free to let us know. So and our team always happy to help with that. So I give some minutes for people to do some survey. And for sure, we have on the resources from on the recording for the single cell exploration club, and also the tutorial and the database doctor data here. The some database is publicly available, and also we also have the SAC club every Friday, so a different time zone. So we feel free to register and join the club. Yep. So I think for that, I would like to say thanks for everyone for joining the webinar today, and hope this information will be helpful. And please feel free to reach out to us using the email like support@bioturin.com for any requests or any discussion that you're interested. Yeah, thanks so much.